Welcome to the first annual Symposium on Mitochondrial Health, Healthspan and Aging, Regulation of Oxidative Capacity through Privileged mRNA Translation by Phillip Dumesic.
All right, thank you. Thank you, Cindy, for the invitation today. I'm thrilled to learn from my fellow speakers about new physiology, new insights into mitochondrial biology. And hopefully what I'll share today, some ongoing work from my own lab, will likewise be of interest to you. So what I'd like to speak about today in particular is a project that we started hoping to study mitochondrial biogenesis and understand how different organisms and different tissues define the number of mitochondria they contain and the activity in those mitochondria. And as I hope to convey, this led us to some surprises and some new signals we think that mitochondria send out that can affect metabolic disease and tissue function. So in general, my lab has been interested in the question of energy balance. This is, of course, the balance between the chemical energy we take in from food and the energy we expend in our basal metabolism, physical activity, and adaptive thermogenesis. This process is remarkable in that it integrates across all of our different organ systems, and it's very adaptable, as we know, accommodating lifestyles from the sedentary to the very energy demanding. But perhaps no argument for the importance of energy balance is as strong as simply to observe what happens when it goes awry. For instance, we know that even a small positive imbalance in this equation plays out over time, leading to obesity and its many health sequelae. These conditions I'm highlighting here, oops, I apologize, are some of the great biomedical challenges of our time, with half of American adults expected to be affected by obesity by 2030 and 15% expected to have type 2 diabetes. And as Rob just mentioned, almost 20% of American children are now affected by obesity, which clearly shows that whatever the root causes of this health crisis, we have not yet stemmed them. I also want to point out, although it's not the focus of my talk today, that negative energy balance is also a common cause of metabolic disease. So in cachexia, we know that inappropriate catabolism outstrips nutritional input, leading to wasting of muscle. This condition, of course, is associated with cancer, with heart failure, with kidney failure, and there's no current FDA-approved medications to treat this condition. So this, we also feel, is a great unmet medical need that my lab studies to some extent. So scientifically, how can we approach this question of energy balance? Well, we could look at it from the perspective of any favorite organ system, but our approach has been slightly more reductionistic. We think about energy balance in terms of individual cells and the decisions they make in controlling chemical energy. So how they take it up, how they transform it, how they store it, and how they utilize it. Because in our mind, these individual decisions, which we can follow in lab and wrap our hands around, ultimately sum up to give rise to the overall organismal energy balance. And so some of the questions we're interested in from the cell's perspective are, what are the programs of energy metabolism that go awry to bring about metabolic disease? And then the flip side of that being, can we reinstall or reinstate adaptive programs of energy metabolism in order to prevent or treat conditions like obesity, diabetes, sarcopenia, or cachexia? So, of course, because of today's theme of mitochondrial biology, I'll talk about one particular adaptive program, and that of oxidative metabolism. Rob introduced that well already. But I just really want to underscore the adaptive or the salutary aspects of oxidative metabolism. And here's just one very stark example, and any sushi lovers in the audience, I'm sure, will appreciate this example. On the right is the muscle of Atlantic bluefin tuna. This fish has extremely oxidative, mitochondrial-rich muscle that can highly efficiently convert chemical energy into physical work to power this fish's migration across the Atlantic Ocean each year. And so this is kind of a mimic for an oxidative, slow-twitch, anti-atrophy program in human muscle that we want to understand how it's built at the molecular level. And in contrast, here's a related fish, the mackerel, that clearly has a grossly distinct oxidative phenotype, a more glycolytic muscle, and one that actually we can't explain from the genomes. The full genomes of these fish have been sequenced, but we can't infer from that. We don't understand how the tuna programs for this oxidative metabolism, and its related fish does not. So these sort of questions are what motivate us in lab to understand how genetically we encode these powerful programs of oxidative metabolism, and as a corollary, how might we be able to manipulate those to treat disease. So this is a project I'll now describe, motivated by that question, that began when I was working in Bruce Spiegelman's lab at Dana-Farber in Boston. And one of his major contributions was to clone a gene called PGC1 alpha. This is a transcriptional coactivator that coordinates a broad gene expression program that helps cells build mitochondria. And so in the about 25 years since the cloning of this gene, it's now well appreciated that PGC1 alpha is one of the dominant controllers of the mitochondrial content of cells in all the tissues of our body. And so we reasoned that if we could understand the signals that turn on or off PGC1 alpha, we might have major insight into how cells dictate their oxidative capacity. And so I'll summarize this first work. It's a little older now, so I'll just show one slide on it. And our major contribution here was to understand that PGC1 alpha is regulated strongly at the level of its mRNA translation. So in other words, the process that converts the mRNA into an active protein. And so what we discovered in particular is that in this 5' untranslated region at the start of the PGC mRNA, there's an additional coding region called an upstream open reading frame. And surprisingly, when we look at where ribosomes are beginning translation on this mRNA, we see they're not beginning on the PGC gene body itself, but rather at this upstream element. And we could prove in assays like in vitro translation assays that in fact this uORF is a repressive element. It's grabbing onto and engaging ribosomes before they can get to the start of the real gene body of PGC1 alpha, and thereby suppressing PGC1 alpha translation. We were interested in how biologically significant this might be, and so we traced its evolutionary conservation. And surprisingly, we found that this 5' leader sequence, or the UTR here, of the PGC gene is one of the most highly evolutionarily conserved 5' UTRs in the human genome. So among our 20,000 or so genes, there's great evolutionary conservation right here, implying significance of this region. So we were able to then trace upstream open reading frames through orthologous PGC1 alpha genes down to fish and even fly. And we could use model systems to then test whether the uORFs are repressive in all these different respective species, or I shouldn't say all these, but a select few of these species. And so here's one example in zebrafish, where we built a reporter system where we could measure translation driven by the zebrafish 5' UTR sequence of its PGC gene. That's read out in the green fluorescent protein expression, and we could see that if we mutate the uORF in the zebrafish leader sequence, we get greater protein output. So the zebrafish uORF is repressive, just like the human one is. Interestingly, in tracing this evolutionary history, we found not only conservation of the uORF, but also examples where the uORF had been lost. So here I bring back the tuna. It turns out that the lineage of the Atlantic bluefin tuna has lost naturally its upstream open reading frame. And so this might be, we hypothesize, a contributor to the highly oxidative muscle phenotype. So here we can test this in our zebrafish system at least. We compare the wild type zebrafish leader sequence with the tuna leader sequence and see that the latter drives much greater translational output than the former. So this is consistent with what we think is an exciting idea, that this translational regulatory element, the gain or loss of this element, is grist for evolution of distinct metabolic phenotypes. So it's an exciting idea, but admittedly this is all correlative, and so we did the gold standard experiment, which is to take a mouse and then experimentally alter the genome just to manipulate this start codon of the uORF. So all we've changed in this mouse is mutating the start codon here. We bred that to homozygosity. I was hoping a tuna would pop out, but we didn't quite get that result, unfortunately. But we could see that in multiple tissues of the mouse. We looked at, for instance, muscle, heart, fat. We see elevated PGC1 alpha protein content. Those tissues are capable at baseline even of higher respiration. They have greater oxidative capacity when stressed. And then significantly, if we put these mice through certain metabolic insults that are known to tax the oxidative capacity of certain organs, we see protection in this mouse. So for instance, if we experimentally induce ischemic kidney injury, the recovery from which is known to depend on PGC1 activity in the kidney, we see protection in our mutant mice as compared to control. So we were quite excited by this. We think it shows that this discrete element in the genome, this translational regulatory element, suffices to control oxidative capacity across multiple tissues and also to influence the course of metabolic pathophysiology. So we wanted to double down on this insight that mRNA translation is a key regulator of PGC1 alpha by searching for more dynamic ways that the cell could alter this gene expression. So the way I've drawn the uORF here, it's built into the message. It's a so-called insys regulator. And so it's somewhat hard to imagine how the cell might dynamically change PGC translation if it wanted to in physiology. And so we set out to look for mRNA binding proteins that might dynamically alter PGC translation. This is a common way that cells confer dynamic regulation to mRNA translation. And we turned, in this case, to a fat system. Fat cells offer certain advantages for searching for new genes of interest. And so I'll briefly introduce this. And suffice it to say that, like muscle, where we can compare highly oxidative to less oxidative phenotypes, and there's a fascinating physiology there to use as a foundation for our research. Likewise, in fat, there's a similar dichotomy, and this is between a highly oxidative type of fat called thermogenic fat as compared to classical white fat. And let me describe that for a moment. So fat has this fascinating physiology that in small mammals especially, exposed to the cold, the fat transforms to increase the number of thermogenic adipocytes. These cells are packed full of mitochondria, and the mitochondria are crucial for burning chemical energy. And in fact, they house specialized enzymes that intentionally run futile chemical cycles in order to dissipate that chemical energy as heat. And for small mammals especially that have difficulty maintaining body temperature, this heat becomes an important defense against losing that body temperature or dropping that body temperature. And you can even grossly see this transformation in the fat, because if you dissect a mouse, for instance, pull out the adipose tissue after cold exposure, you see this starkly brown coloration of the fat, reflective of increased blood supply, increased iron, increased mitochondrial content in the cells. So as you might imagine, PGC1 alpha, because it programs cells to build mitochondria, it's a crucial component of thermogenic fat because you need these mitochondria to burn all the chemical energy that provides the heat. And furthermore, PGC1 alpha directly activates certain genes, including the so-called uncoupling protein, that are key to run these futile chemical cycles that are ultimately important for heat production. So just as we talked about the mackerel versus the tuna, we can use comparisons between thermogenic adipocytes and classical white adipocytes that aren't as metabolically active. They function rather to store chemical energy as triglyceride, when we can use this sort of contrast to better understand how cells define the program of oxidative metabolism. So I should also say, although my lab does basic research, we're of course interested in human health and how to treat it. And there's an emerging idea about using or leveraging the physiology of thermogenesis in order to treat metabolic disease, because as I described, it's a fundamentally calorie burning process that could, in theory, confer benefits to energy balance. And so about almost 20 years ago now, this idea took hold because it was demonstrated that at least some humans maintain pockets of active thermogenic fat. These tend to be subclavicular. That's what you can see here in the PET scan. And there's a correlation. Those who maintain these pockets of active thermogenic fat tend to have improved cardiometabolic parameters like improved blood glucose levels and improved blood lipid profiles. And so this has, like I said, raised the possibility or the idea of somehow activating this physiology for therapeutic gain. However, there are major obstacles to that goal. So first, compared to mice at least, on a pound-for-pound basis, we simply don't have that much thermogenic fat. So the question remains, even if we had a molecule that could activate these cells selectively, would that be enough in a typical human to move the needle in physiology? And so this problem, or this obstacle, has led to a lot of interest back on the classical white adipocyte. These are the main types of adipocyte in our bodies, and we wonder if we could coax more thermogenic activity or metabolism out of these cells. That may be a more relevant or abundant therapeutic target than the thermogenic adipocytes per se. And furthermore, another obstacle is the inflammation that's associated with obesity. So as we know, there's chronic low-level inflammation in multiple tissues that accompanies obesity. And classic observations for decades now establish that this inflammation prevents PGC1 alpha expression or dampens PGC1 alpha expression and dampens mitochondrial biogenesis. So we think that the patients who struggle with obesity, who would most benefit from the activation of thermogenic fat, have this additional obstacle of the inflammation in their adipose tissues. And I want to emphasize that this observation that inflammation suppresses mitochondrial function and biogenesis, it hasn't just been made in thermogenic fat. This is also seen in white adipocytes. And I want to be clear, these also express PGC1 alpha just at a lower level than do thermogenic adipocytes. So these red arrows, these repressive arrows, have been of intensive scientific interest for decades now, because it's been proposed when this was first observed in mice and in human twin studies that there's this decrement of PGC1 alpha, decrement of mitochondria in obesity. It was proposed that a sort of metabolic insufficiency or mitochondrial insufficiency might push one over into type 2 diabetes or lead to the pathogenesis of that condition. However, it's always been mysterious, and this point is still debated. It's been mysterious what exactly the insufficiency is. So as I described these white adipocytes, they function mainly to store chemical energy. So why would a slight quantitative decrease in mitochondrial content or respiratory capacity be so problematic? If these cells don't have to do much respiration, why would it make such a difference for there to be a slightly smaller level of mitochondria? So that's been, to me at least, a mystery, and I'll come back to this question as we proceed. So again, our initial basic science motivation here was to understand what are the genes that the cell might use to dynamically regulate the translation of PGC1 alpha. And so we sought to find mRNA binding proteins whose loss changes translation of the PGC1 alpha gene. And so we did a candidate-based screen in adipocytes where we knocked down individual mRNA binding proteins and looked at PGC protein output as a readout of PGC translation. And our top hit in this small screen was an RNA binding protein of unknown function called RBM43. And what I'll emphasize here is that the directionality of this result. So when we knock down or when we get rid of RBM43, we see an increase in PGC1 alpha protein. So RBM43 looks like a repressor of PGC1 alpha expression. And part of the reason RBM43 was in our short list in the first place is that its expression pattern is quite distinctive. It is more highly expressed in white fat than in thermogenic fat. And when mice go in the cold, so in other words, when they need to turn on thermogenesis and turn on that oxidative program, RBM43 expression goes down. So its expression is inversely correlated with respiration. And so it makes sense then that when we get rid of RBM43, we see an increase in PGC1 alpha. And then finally, we are interested to pursue this top hit of the screen because there were human genetics data linking RBM43 to metabolic disease, specifically type 2 diabetes. And this signal is especially strong for reasons we don't understand in the African American population. So I'll now show some raw data from the screen. So this is two different small siRNAs to knock down RBM43, and each increase the PGC protein output with not a commensurate change in the PGC mRNA. So this looks like potentially a translational output that's been affected. We also don't see a change in the PGC protein half-life, again, pointing to mRNA translation. But to more directly assess that, we could do something called an in vitro translation extract, where we grind up a cell and use its ribosomes to translate mRNAs that we synthesize experimentally. And so here what we do is we can take We can take the untranslated regions, so the regulatory elements of the PGC gene, and stitch it to a luciferase reporter gene. And what we see here is that when we make translation extracts that have excess levels of RBM43, that suffices to reduce translational output of this PGC reporter sequence. So just putting the 5' UTR of the PGC gene on this reporter now confers the ability to be repressed by RBM43. And we see if we mutate the uORF, that has no effect. So this seems to not involve the uORF that I described before. It's some other element in the 5' untranslated region, confers the ability to be repressed by the RNA binding protein RBM43. So now, if we get rid of RBM43, we induce PGC1 alpha. We've been talking about PGC1 alpha as a determinant of mitochondrial function and biogenesis. So do we, in fact, then see an increase in mitochondrial capacity? Well, we do. So these are the adipocytes where we've knocked down RBM43, and they express higher levels of these genes and enzymes important in the mitochondria. And when we measure respiration directly by measuring oxygen consumption, we can see that the RBM43 knockout cells are capable of higher levels of respiration. So indeed, it seems like we've engaged that PGC1 alpha downstream program. And so we went ahead and made a mouse model. So we used some genetic tricks where we could knock out the RBM43 gene selectively in adipocytes. So this mouse is totally normal except for its adipocytes that lack RBM43. And what we see, as expected, is that the adipocytes now express a greater level of the PGC1 alpha protein. They haven't changed the mRNA level of PGC1 alpha, but they've increased the protein output, we think, because of a change in mRNA translation. And we can look at that reasonably directly by doing a trick. So we can take the fat tissue from mice. We can leverage the buoyancy of the fat cells to float them up to the top of our buffer, and while the other cells get pelleted down to the bottom. And then we have those pure adipocytes, and we can look at where the ribosomes are translating different mRNAs. So we basically use a sucrose density gradient to separate highly translated polysomes, mRNAs covered with ribosomes, from the low-density fractions where you have mRNAs that are not associated with ribosomes. And what you see is that the PGC mRNA in particular, when you knock out RBM43, it now shifts to the higher density fractions, indicative of greater translation, more ribosomes on the PGC mRNA. And we think, therefore, that greater translation is contributing to this increase in PGC protein in our mutant mice. So what does this do in the fat? Does it, in fact, engage mitochondrial biogenesis? Well, it does. So when we use proteomics to look at about 5,000 or 6,000 genes across the proteome simultaneously, we see that many mitochondrial proteins are elevated, so about a 25% increase on average of mitochondrial proteins. We think that's reflective of just a greater mitochondrial content in these cells. And although this upregulation is modest quantitatively, I'll remind you that in these classic obesity studies I cited, identical twin humans, lean versus obese, mouse studies as well, it's typically about a 50% change in mitochondrial content comparing lean versus obese. So we think this 20 to 25% change we're seeing could be certainly physiologically meaningful. And so importantly, what we can do here also is a cool trick. We can take our RBM43 fat selective knockout animals and breed in one allele of PGC1 alpha that can be knocked out as well. So what this does in effect is it lowers the PGC1 alpha back down to baseline levels. So we're still knocking out RBM43, but now we're normalizing PGC1 alpha levels, and that brings the mitochondrial content back to baseline here. So we really think that RBM43, therefore, is acting through PGC1 alpha in order to control mitochondrial biogenesis. And in theory, oxidative metabolism, although I haven't showed you that yet, so I'll show it here. These are more directly metabolic studies. What we can do is dissect the fat and look at the minced tissue in a respirometer. We see that it indeed engages in higher basal levels of respiration. We can also do a more elegant experiment where we have living mice individually housed in cages, and we use what we call clams cages to, in real time, measure the energy expenditure of these mice. And so what you can see here is if we inject the mice with either a control, just salt water, we get kind of a stress response that's no different between the RBM43 knockouts and the controls. And in contrast, if we inject a beta-3 receptor agonist, this induces thermogenesis on adipocytes selectively because that receptor is expressed selectively in adipocytes. Here you see the stress response, but you have this longer tail of energy expenditure that represents, we think, an increased capacity for adipose thermogenesis in these knockouts as compared to wild type. So it looks like the RBM43 knockout animals have greater mitochondrial content in the fat, greater capacity for adipose thermogenesis and respiration. Okay, so what might this mean for metabolic disease? Well, we were quite interested as we set up our cohorts of mice to do longer-term feeding studies, quite interested to realize that the promoter regions for both mouse and human RBM43 genes are decorated with potential binding sites for important transcription factors involved in inflammatory signaling. This includes interferon regulatory factors and NF-kappa B. And so this was interesting. It suggested that RBM43 may be induced by inflammation. And sure enough, if we take isolated fat cells and treat them with certain cytokines or activate innate immune sensors in these cells, all of these stimuli induce, sometimes quite strongly, RBM43 expression. So it looks like inflammatory signaling is one input into RBM43, and then as we've shown, we think RBM43 then suppresses PGC1 alpha and oxidative metabolism. So how can we test this now expanded working model? Well, what we can do is, again, treat adipocytes with cytokines, but now either knock down RBM43 or not, and see if we can interrupt this axis. So we know that treating adipocytes with cytokines dampens respiration and dampens mitochondrial function. And you can see that here by comparing the gray trace here to the blue. So a TNF-alpha, this classic cytokine, if you treat adipocytes for 24 hours even, you get this stark decrease in mitochondrial respiration. And in contrast, if you get rid of RBM43, you're now protected from that TNF-alpha-induced drop in respiration. So this to us suggests that indeed there's an axis here where inflammatory signaling acts at least in part through RBM43 to repress oxidative metabolism. So going back to my introduction in those red repressive arrows, we think that RBM43 could be part of the machinery that dampens respiration in the context of adipose inflammation. So this, of course, made us very motivated to put our mice on high-fat diet and watch their weight and their health over time. And as I told you about thermogenesis, if thermogenesis is altered here, that should be burning calories. It should be a negative energy balance and potentially protect these mice from weight gain. In fact, we did not see that. So this was late in my postdoc when we did these studies, and so you can imagine my consternation when we saw that there was no change on a high-fat diet in the body weight or the body composition of these animals. So although I introduced this in terms of thermogenesis, it does not look like the RBM43 knockout animals have a stark difference in energy expenditure. And we could also measure how much food the mice ate. There weren't changes there, weren't changes in how much the mice were moving around. So it did not look like we had a change in overall energy expenditure. However, where we saw something quite interesting is when we performed glucose tolerance tests to look at the glucose control by these animals, we see that the RBM43 knockout animals are strongly protected from the glucose intolerance that comes with obesity. So these mice, again, they are obese just like the controls, same accumulation of fat mass, but they're somehow protected from one of the key metabolic consequences of that, weight gain. So this phenotype, you may be aware, is classic for alterations of adipose inflammation. It's been observed many times in the past that if you alter a mouse such that you block inflammation in the adipose tissue, you get this sort of phenotype, same weight gain, but an improved systemic glucose control. And indeed, what we see in our RBM43 knockout animals unexpectedly is that they were protected from some of the macrophage accumulation and inflammation in the fat that accompanies obesity. So now, to some extent, we were back to square one. We said, well, how had this intervention we made that alters PGC1 alpha and mitochondrial biogenesis, why had it unexpectedly affected inflammation? We were thinking initially in terms of just tabulation of energy balance. So we turned to the visceral adipose depots, now in our high-fat diet-fed animals, and tried to characterize them in an unbiased universal manner. So again, we confirmed that PGC protein levels are up in our knockouts with no change in the mRNA. So that's, of course, consistent with the change in protein translation. And then we used proteomics to, again, assess across the proteome any quantitative changes. We see our top upregulated term is oxidative phosphorylation. That makes sense. We're seeing, again, the mitochondrial biogenesis program engaged here. But interestingly, we now see this broad array of downregulated terms in our knockout animals, and many of them relate to inflammatory responses that have been decreased. Also, if we use Evan Rosen's signature of high-fat diet adipocyte gene expression, we see that this signature associated with obesity is profoundly down in our knockout adipocytes as compared to controls, much more so than any individual program. The kind of broad obesity signature is down. And I'm showing some examples of famous genes that are part of this signature. These include genes that are proteins that are secreted by adipocytes and thought to alter the adipose milieu as part of the pathogenesis of obesity. So the punchline here is that these adipocytes, the RBM43 knockout adipocytes, they're in an obese mouse, but somehow they don't feel obese. They haven't engaged some of these programs that remodel the adipose in likely damaging ways in obesity. So why is it? What upstream pathway, presumably, has changed to affect so many different downstream pro-inflammatory programs? So to try to figure that out, we looked at not only the most changed proteins in our proteomics data set, but we also used several unbiased tools to try to predict signaling pathways that were different in our knockout cells versus wild type. And those two strategies converged on a very interesting protein I'll describe now called STING. So the STING protein is reduced strongly in the RBM43 knockout adipocytes, and in informatics, signature of STING activity is also down in the knockout adipocytes. So what is STING? Well, STING is part of an innate immune sensor pathway that helps cells detect when a pathogen is present. So this sensor detects DNA that's inappropriately in the cytoplasm. The sensor is called cGAS, and that synthesizes a cyclic dinucleotide second messenger, which ultimately activates STING to serve as a platform for several signaling cascades that ultimately turn on pro-inflammatory gene expression driven by IRF3 and NF-kappa B. And I highlight those because, again, we saw both of those programs regulated in our knockout adipocytes. And so this would be a potentially explanatory upstream regulator that could touch both of those pathways. So, okay, so pathogens, how does that make sense in the context of obesity? Well, a recent idea, not originally from us, but one we've latched onto, is that in obesity, there can be observed in several cell types an increase in mitochondrial DNA that's inappropriately in the cytoplasm. So not a pathogen per se, but DNA derived from our cells from the mitochondria that's now inappropriately accumulated in the cytoplasm. It's then detected by cGAS as if it's a pathogen to trigger this pro-inflammatory cascade. And so we saw these hints in the literature. We sought to replicate it ourselves. And indeed, we see that in our high-fat diet-fed mice, there's an increase in mitochondrial DNA that's inappropriately in the cytoplasm of adipocytes. That corresponds to an increase in this second messenger here. And then furthermore, using immunoblots, we can document these signaling pathways downstream of STING and see that they're induced in obesity, as had been reported before. And then the surprise here, the more significant observation from our project standpoint, is that in our RBM43 knockout animals, they were strongly protected from this obesity-associated activation of STING signaling. So we didn't see the cytoplasmic mitochondrial DNA accumulate, we didn't see the second messenger accumulate, and we saw that most of the obesity-associated STING signaling is prevented in the context of the RBM43 knockout. So RBM43 is unexpectedly a regulator of this innate immune signaling pathway in adipocytes. And so we wondered, well, could this have something to do with PGC1 alpha as well? In data I'm not showing you, we think most of the effects of RBM43 depend on PGC1 alpha. So could it be that PGC1 alpha, this biogenesis regulator, is unexpectedly regulating innate immune signaling. And so that's, in fact, what we see. So to test that, we now turn to adipocyte systems, and we just knock out PGC1 alpha to confer or enforce a state of PGC1 alpha insufficiency. And it turns out what happens is then you have fewer mitochondria. You can't do biogenesis as well. You have less total mitochondrial DNA. But more of that mitochondrial DNA is inappropriately in the cytoplasm where it shouldn't be. And you set off this STING signaling that we can observe here by immunoblot. And then perhaps even more importantly, we can show at the gene expression level, when you're insufficient for PGC1 alpha, when the adipocytes are, they induce these pro-inflammatory genes activated by STING, and we can really prove that by treating these cells with a chemical inhibitor of STING called H151. And when we do that, we completely suppress the activation of these pro-inflammatory genes. So unexpectedly, PGC1 alpha is not only playing a role in building mitochondria and conferring that capacity for oxidative metabolism, but it's also a safeguard against the accumulation of this danger signal, cytoplasmic mitochondrial DNA. And we think in doing so, PGC1 alpha is a safeguard also against adipose inflammation and some of the negative metabolic consequences of that adipose inflammation. I want to be clear, we haven't yet, we're very actively doing this. We're now knocking out STING to prove that it's really STING signaling that's driving this adipose inflammation. So that part of the arrow here we're still working on, but at the very least, I think we've unexpectedly seen that this regulator of biogenesis is also a safeguard against these important stress signals. So to summarize what I've spoken about today, I told you, with fish as an interesting jumping off point, I told you that translational regulation of the PGC1 alpha gene is an important governor of oxidative capacity in multiple tissues, and we think that's also potentially relevant for metabolic disease. I then told you about this RNA binding protein, RBM43, that represses PGC1 alpha translation. It's the first RNA binding protein proposed to regulate PGC1 alpha primarily through mRNA translation. And I showed you as part of this what we think is diabetogenic signaling axis here, where it represses PGC1 alpha, and that has these two-pronged effects. It limits oxidative capacity, and it also leads to this unveiling of the stress signal of cytoplasmic mitochondrial DNA. And we're also quite intrigued by findings in other tissues, like especially the brain, that show dampening or decrement of PGC1 alpha levels in aging that correlate with an activation of STING in the absence of pathogens. So we think perhaps in tissues even beyond fat, this axis may be important for aging and for physiology. And so finally, I want to end on this question I posed before of this famous observation that there's a decrement in mitochondrial content in obesity in tissues like fat and muscle. What exactly is the problem that causes? And again, much fantastic work over the years has documented very carefully the metabolic insufficiency, the respiratory insufficiency quantitatively that can be observed as that mitochondrial content drops. But what I'll argue today is perhaps it's not the respiration per se that's the key variable. Perhaps another key problem here is the stress signals that are emanating from mitochondria, namely the cytoplasmic mitochondrial DNA. And indeed, if we take human patient samples comparing lean versus obese, or amongst obese patients comparing insulin sensitive versus insulin resistant, we see a STING activity signature correlate with worsened metabolic parameters. So we think it's not just a change in respiration per se, but rather these danger signals that could be especially important for metabolic disease. So with that, I'll stop and acknowledge many fantastic collaborators we've had along the way. So as I mentioned, these projects began in Bruce Spiegelman's lab at my time at Dana-Farber when I worked with Dan Egan and Samantha and Sarah, two fantastic technicians in lab. And then we had many fantastic collaborators across all sorts of model systems, from yeast to flies to tuna fish even. And thank you all for your attention today. I'm happy to take any questions.
Learn more at www.blueoaknx.com/symposium.